Today, basically, my talk will be focused on dung beetles. First of all, I would like to introduce you to the structure. So the most part of the talk will be devoted to dung beetles, their phylogenetic and systematics. And then at the very final moment, I will discuss my present project on modeling evolution using ontologies, which actually the project which I am going to do here. I just started my postdoc at NIMBioS around 2 months ago, so therefore I don't have by now a lot of things to discuss about my ongoing project at NIMBioS. So I'm going to take you to African savannas, and here you can find large herds of herbivorous mammals which produce tons of dung. And this dung is massively populated by dung beetles. And actually these beasts are the main topic of my today's talk. Dung beetles are distributed worldwide, so they number around 6,000 species, and these species are classified in around 200 genera and 12 tribes. And actually, diversity of dung beetles is quite fascinating. They come up in different shapes and forms. I was very curious about dung beetle diversity since my school years. And actually, around 30 up to 50% of dung beetle diversity is undescribed. So I started my career in biology by studying taxonomy of dung beetles, but by basically describing this undescribed diversity. And while doing my undergrad studies, I had really a great chance to go as an exchange student to Laos. And Laos is a country in Southeast Asia. So I spent more than a year collecting dung beetles across Laos. And the diversity there of dung beetles is quite awesome, actually. You never know where you find a new species. So it can happen in any most unexpected moment. And this is actually the first the first species I found in Laos. So the first species to science, the first in my life. So now I'm going to actually tell you a story, how it happened. So I arrived at a very nice collecting site with very nice primary forest here. And then I felt an urge to find a restroom. I found one for myself right in this forest. And then suddenly I noticed a beetle which was flying over me. So in a jump I caught it, and it was this weird looking ontophagus. Compared to other ontophagus, it looks really very weird. And later in the lab I identified that actually this is a new species. So basically the conclusion, it all depends on a good collecting site. But actually there are many other charismatic traits of dung beetles which make them stand out among other animals. So dung beetles are actually popular organisms in biology in general. So for example, this chart summarizes the citation per beetle subfamily. So dung beetles is a subfamily. And actually dung beetles come up the second most cited subfamilies on Google Scholar. And this is not a surprise because dung beetles are actually model organisms in ecology and developmental biology. And I'm going to review it in a bit more detail. So why they are the models in ecology, for example? Because dung beetles, first of all, are quite cheap to collect. So they don't need much money. And it's quite easy to process, quite fast to process them in comparison to other animals. So therefore, dung beetles are used in various ecological surveys and in environmental monitoring. So also dung beetles are model organisms in evolutionary developmental biology. So many of dung beetles, many males in dung beetles primarily, developed various shapes of horns. And actually the genetic mechanisms which underlie this tremendous horn diversity are actually the focus of current evo-devo science. And dung beetles actually seems to be quite promising to resolve current issues in this branch of biology. Why not using? Another example which is worth mentioning is actually dung beetles in Australia. Australia has quite a rich fauna of native dung beetle species. When Europeans arrived in Australia, they actually brought a lot of cattle with them. But native Australian dung beetle species were not capable to utilize this dung, which was produced by cattle, because they actually adapted to feed exclusively on the dung of marsupials, and the dung of marsupials and cattle of herbivores, mammals, is quite different. So, but what started to happen? Basically, uncontrolled no one decomposed that dung, so it stimulated uncontrolled production of pestilent flies, so because they could breed in this dung quite easily. So, and also it caused kind of massive pollution on pastures. So the solution actually was found when Australians introduced dung beetles from Africa to specifically utilize the dung of cattle in Australia. So another case, also a charismatic feature of dung beetles. Dung beetles are only known animals beside humans which use Milky Way to navigate. So, and to effectively use dung beetles as model organisms in biology and ecology, you need to know their phylogeny. So and what's happening in dung beetle phylogeny right now? So when I started this project on dung beetle phylogeny in 2013, by that time there were around 13 major works which were focused on basically higher phylogenetic relationships in dung beetles. And the main consensus that all those 13 works shared in common was the lack of any consensus. So basically phylogeny of dung beetles was unresolved. And I decided to revisit phylogeny of dung beetles by analyzing all possible kinds of data you can analyze in this case. It's anatomy, fossils, DNA. So, and basically this was my PhD project. So, and this will be the main focus of my today's talk. So I will start from morphology-based phylogeny of dung beetles. So when you start studying morphology, so you basically start with detailed investigation of morphology. And morphology is quite variable in dung beetles, so it's quite interesting. So in this morphological survey, I tried to select all major taxonomic and biogeographic lineages in dung beetles. So all in all, it resulted in over 110 species, which I scored for 205 characters. And then I analyzed this character matrix. So indeed, actually, it's quite tedious work to code morphology. So it took me around, I think, 2 years to score these matrix, just 110 species and 205 characters. So because you need to conduct multiple comparisons between different species. So, and I analyzed them using conventional paradigm, it's parsimony, which is used in the analysis of morphology characters, and also Bayesian approach. So the Bayesian approach was mainly employed to see if Bayesian framework can give any new insights into analysis with morphology. So here is the parsimony tree. So actually what is good about parsimony is what is absent actually in Bayesian or likelihood framework when you do an analysis of morphology is that actually you can, there are techniques in parsimony which allow identification of some potential characters which introduce phylogenetic noise. So you can eliminate those characters and get, for example, better resolution of a tree. So that's actually what I was trying to do here. So that's the tree itself. And then Bayesian analysis. So as I mentioned, actually the main idea was to test whether Bayesian analysis can give any new insight. And this was actually mainly performed by partitioning morphological data set, because partitioning of data set allows different sets of characters to evolve at different rates. And while partitioning in molecular framework, when you analyze molecular data, is more or less straightforward, so you have different genes or codon positions, so in morphology it's not. So here I partition dataset based on the assumption that characters of the same topological location are more likely to evolve at similar rates. So basically the characters of the same organ tend to evolve at more similar rates. And to do that, to perform this partitioning, I represented the anatomy as a tree structure, as anatomy ontology. So basically this tree structure reflects the relationships among different sets of organs. Like for example here, these are characters of mouth parts, and these are characters of the rest of the head. So together they constitute a bigger clade which corresponds to the characters of the head. So basically you can use this tree and you can navigate through this tree from the tip to the bottom and merge sister nodes to come up with different partitioning schemes of morphology. So it can be one partitioning scheme which treats all the characters as the same partition, or it can be multiple, like in this case it was up to 11 different partitioning schemes where all elementary blocks, the tip blocks, are treated separately. So, and then such program as NIMBioS, for example, it allows linking and unlinking parameters among different partitions. For example, among character rate variation, among partition rate variation can be linked and unlinked, and this allows modeling different sets of morphological characters to evolve at different rates. Here, so I think, yes, I came up with so all combinations of partitioning schemes and parameter linking schemes actually resulted in 82 different Bayesian analyses. So, and this Bayesian analysis can be compared using marginal likelihood of Bayes factor. So, and the main results of this analysis, their model likelihoods are shown here. So the main conclusion is that more complex models where you have unlinked parameters and morphology which is partitioned, so actually tend to have higher likelihood. So basically given the fact that actually they have sort of perform better. So, but actually despite variation in marginal likelihood, the topologies of all analyses were quite similar. So basically it doesn't matter whether you use partitioning or not, you get quite similar results. And well, if you focused on just finding topology, so for example taxonomic purposes or whatever, then I think one parameter scheme which uses only one partition is actually the best because it saves time to compute all these analyses. However, if you are interested in actually at the accuracy of parameter estimation, then partitioning of morphological data set actually can be quite meaningful. So with higher likelihoods, you are more probable to get better parameter estimates. So, and here are the results of those parsimony tree from morphology and Bayesian tree from morphology. So parsimony tree is better result, but actually, despite these resolution differences, they quite congruent, both of them, Bayesian and parsimony. So, and this is actually current taxonomy, which is mapped onto the tree. So as I mentioned, there are 12 tribes in dung beetles, and all of them came up monophyletic here, or like, yeah, almost monophyletic. So, but there are 3 tribes which always come up highly monophyletic, highly polyphyletic. So they spread out across the tree. So basically, those tribes are artificial. Their tribal concept need revision. And here are the results of parsimony tree, and actually here I mark that those clades which were consistent with the previous molecular analysis. So however, as you can see, the bunch of the tree is still inconsistent, and actually in those molecular trees also the big parts of the trees are not are inconsistent with each other with this phylogeny. So here I'm going to move to another. So yeah, and the next step in my project, in my PhD project on the dung beetle phylogeny was actually accumulating of molecular data and analysis of molecular data. So let's get back to the history of research in dung beetles. So actually, as I said, there were 13 major works, and actually 7 of them were molecular phylogenies. And the case with molecular phylogenies was even worse than with morphology, because all these phylogenies were quite conflicting with each other. In my phylogenetic work, and basically all of those phylogenies, they were based on 1 or 2, rarely 3 genes which were used to infer phylogenies. So basically, in my molecular project, I was trying to increase the gene sample by sequencing six gene regions and by accumulating global sample of dung beetles. So there were lots of sequences already available on GeneBank, and I sequenced another 100 taxa, which covered previously not included genera and species. And then actually I was trying to select molecular data using Bayesian posterior prediction. So what does it mean? So basically, statistical models we use for inference are imperfect, but normal optimality criteria we use to infer the best models, like for example, archaic information criterion or Bayesian information criterion, are relative measures. They just show that some particular model is better than the other model. So, but they do not really show how best this model explains the data. So, and the way to deal with that issue is actually to use Bayesian posterior prediction. Bayesian posterior prediction can specifically answer questions how adequately a model you use reflect the processes which generated the data. And the analytical procedure which I employed was the following. So, Bayesian posterior prediction works by taking you take your original data, then you do Bayesian inference, and you get a set of trees and parameters. So, sample of posterior distribution, and then can use this set of trees and parameters to simulate data, to simulate molecular data in this case. And then you can compare how the simulated molecular data are similar to the original data. So there are actually not so many programs which can deal with posterior prediction in molecules. So what I did in my molecular metrics, so I was trying to partition every gene using either codon position or domain structure or secondary structure as in RNA genes, and then trying to infer their phylogenies and parameters using Mr. Bayes. And then there is a Puma software which actually makes all these predictive simulations and compares the simulated data using the parameters from posterior sample with the original data. So, and here you can see this graph which summarizes all the results. So it's different genes, and the genes were partitioned themselves. So, and Puma gives a p-value. P-value is a kind of statistics which called multinomial likelihood. So if your multinomial likelihood falls in the center, it means kind of your data or the model you use to analyze this data is good. If the multinomial likelihood falls in the tails, then it means like it doesn't really explains the processes which generated your data. So actually what is interesting that majority of these partitions have kind of quite okay p-value, and specifically RNA genes, specifically 18S, has actually the highest p-value. So which can be used to say that, yeah, basically the models we used to make our inference actually explains explained data more or less okay. Then I was trying actually to use this p-value to exclude those partitions where p-value was not good. So, and then I was trying to analyze this character data, molecular data set using different analytical procedures, Bayesian, maximum likelihood, and parsimony. So, in general, all analysis is except parsimony using dynamical approach. So actually yielded quite similar topologies. So, and the main result of this, I will show you the tree later. So the main result that actually 4 tribes here were found to be highly polyphyletic, while the rest of the tribes were quite monophyletic. So, and you can see this tree here. So those 4 tribes which are found polyphyletic, indeed actually their monophyletic concept, which was found in this analysis, is consistent with the previous morphological analysis. So, and the congruency between morphology and molecules basically provided quite good support to define new concepts for these tribes, basically to reclassify the existing tribal concept in dung beetles. So, and this is actually what was done by me and my collaborator. So we established new concept for those most polyphyletic tribes in dung beetles using molecules, using a consistency between morphological and molecular analysis. So, but that poses another problem, because we classified those polyphyletic tribes. The rest tribes were monophyletic. But by reclassifying those polyphyletic tribes, around 100 genera remained unclassified. So they now currently they do not belong to any known tribes. And this means that actually many new tribes, given my brief estimate, around 50 new tribes have to be established in dung beetles. And this consistency between molecules and morphology also tried to solve another problem in classification of oriental dung beetles. So there were 2 genera in oriental region of dung beetles. These genera have never been deemed to be closely related to each other, and their tribal placement was always unstable. So different authors placed these genera into different tribes. And then we found actually a new species in Laos, which gave a hint that probably these 2 genera are closely related to each other, because it was a kind of morphological link between those 2 genera. And actually I tried to test this hypothesis using morphology, so by integrating the representatives of those genera into the morphological character matrix, where they actually came up monophyletic by forming a monophyletic group which is separated from all other genera. And actually molecules also confirm that. In molecular data set, they also come up separately, so by forming a separate clade. So basically these allow to put these 2 genera, to first of all synonymize them, because they're basically the same, they're not different genera, it's the same genus. So, and to assign them this new genus, the new concept of this genus, into a new tribe, which is called Paracarini, and this tribe I revised. It turned out that actually many species which are found and which belong to this tribe actually are new to science. So this tribe is exclusively endemic to Oriental region. So, and actually one of the last category of data which remained poorly explored in dung beetles when I was doing my PhD, so actually were dung beetle fossils. And fossils are important to judge timing of dung beetle evolution, actually to conduct total evidence analysis, to incorporate them with the extant species. So what was interesting about dung beetles fossils, there were not so many fossils now, and it's around 31 fossils because of their lifestyle, so associated with dung, which is not fossilized. So, and their oldest fossil was dated at Upper Cretaceous, me and my collaborators, we revised these fossils, and actually it turned out that only 21 fossils can be reliably assigned to dung beetles. All the rest are not dung beetles. And actually the oldest known fossil, which was dated at Upper Cretaceous, is actually also not a dung beetle. So that actually shifted the minimal age of dung beetles not from Upper Cretaceous to Eocene. And we also integrated those fossils, the best preserved, into character matrix, and now they are available for total evidence analysis. So basically, the main conclusions from what is done right now in systematics and dung beetles, in the phylogenetics of dung beetles by me and my collaborators, is that morphological and molecular phylogenies corroborate each other, although there is some discrepancy, especially what concerns intermediate divergences which are poorly supported. And present classification has to be substantially revised, so many new tribes and dung beetles have to be described. And now I'm going to give a review of my ongoing projects. So yes, and dung beetles are still in process. This is my current ongoing project where we're trying to actually finally integrate all those kind of data, DNA, anatomy of fossils, in the framework of total evidence analysis to get a plausible data tree of dung beetles. So then some other of my ongoing projects. So as I previously mentioned, some dung beetles have horns. Male of dung beetles have horns. And given current theory, those horns are used to fight for females or block tunnels, because dung beetles which have horns are tunnelers. They live in tunnels underneath dung path. But actually, it turned out that there is a poorly known group of dung beetles comprising around 5 species and occurring in China, which have movable horns. So, and to me, actually, it's the only animals on Earth which have movable, to my knowledge, which have movable horns. So basically, the horn, So how it works, how this connection works. The cuticle, where in the place where horn merges clippers, is thin and elastic. So this allows, and there are muscles which join, you can't see it here, but they are. So we join horn and clippers. So this allows movement of horns in anterior-posterior direction. And actually, it's quite interesting because these species are poorly known. This construction of movable horn contradicts kind of current theory why horns are needed, because you can't fight for females, or you can't block tunnels using such movable horns. It's just very weak to do like any forces in the rivals. So me and my collaborators now are trying to understand what these movable horns evolved for. So, and now finally, I'm going to tell you about my postdoctoral project, which is my main project presently. So this project is focused on using ontology-informed models to reconstruct evolutionary dynamic of entire phenotype. So current phylogenetic methods basically reconstruct evolutionary dynamic of a particular trait or character in a trait-by-trait manner. So they basically reconstruct evolution dynamics for each character separately. But thanks to works of morphologists, so we have big character matrices for different groups of animals right now. So basically have good phenotype data, but there is no method right now in statistical biology which can take and model all those data at once. On the top of that, actually, anatomical characters bear some dependencies, which we call anatomy ontology. So these dependencies is very easy to comprehend. For example, like eyes is a part of head, or tarsi is a part of tibia. So all of these dependencies can be represented as a network, or in a very simplified way, is as a tree here. So, and this tree, so this big network evolves, this big network of phenotype evolves over the course of evolution. For example, and these dependencies are important. For example, we have lizards which lack limbs. So as soon as limb disappears in the course of evolution, all digits which are located on the limb also disappears. So therefore, the dependencies which are given by this network have to be taken into account when you model phenotype evolution. So, and actually, that's what my postdoc is going to be focused on. And the model which can be used here is actually, well, the statistical framework is actually the framework of Bayesian networks. Bayesian networks are frequently used in medicine, where they employ to understand the causes of different diseases. And Bayesian network is basically an acyclic graph, which represents the conditional dependencies of the nodes, between nodes. So basically, this framework is very, very convenient to model those dependencies which exist in anatomy ontology in the phenotypic network. And this way of modeling actually also changes our present kind of tradition how to model phenotypic characters, because using this approach we will be able to understand how rates of evolution in different systems of characters and different system of organs evolve differently or in the same manner. So we will be able to understand the heterogeneity between them. And this has the potential to answer some key questions in current biology. First of all, it's adaptive dynamics of morphology over space and time. It's evolutionary dynamics of morphological complexity, and the origin of evolutionary novelties. So, yes, and in my project, I'm going to test these models using wasps and dung beetles. So that's basically all. Thanks for your attention, and I would like to acknowledge many of my colleagues listed here with whom this research won't be possible. And yeah, I would be glad to take your questions if you have any.
Start if no one else. Go ahead. Oh my. Okay. This is a question about the term phenotype, because for your project, you've used this in a way that I think is a bit more general than morphology and the genetics, potentially. And I'm asking because Kimberly is here, and there's a whole set of behavioral and or ecological syndromes associated with potentially different species. Has anyone, or do you, plan to sort of take those into account in doing a phenotypic analysis? Okay, because what you talked about was a phenotype analysis, not genetic. And or is this totally crazy?
No, well, here I understand. Of course, I assume that just in my talk here, phenotype is just array of characters, of morphological characters, so by anatomical characters. But you can, I think, extend it to behavioral traits as well. So just a matter how to model that. Yeah.
But is there anything really different about that? So, for example, plasticity of behavioral characters may be much broader than plasticity of morphological characters for a particular genotype.
Maybe. So,
Maybe.
and here, yes, maybe I didn't mention that, but I'm going to focus specifically on discrete traits, on discrete characters.
Okay.
So, well, you can also extend it to a continuous part, actually. These dependencies, they appear mainly in discrete. Well, if you, yeah, of course, they also appear in continuous ones, but this is the main focus so far, because I think in the future you will be able to extend these models, but yeah, behavioral traits can be included, I believe.
Okay, great, thanks.
Hello? Okay. So you mentioned about dung beetles using the Milky Way to navigate. Can you expound upon that?
What's it, sorry?
You said that dung
Yeah.
beetles use the Milky Way
Mhm.
to navigate. Can you expound upon that?
Expound what?
Can you say more about it? How to use the Milky Way?
Actually, it was discovered not very long ago, so I think it's 2013 paper. So, yeah, exactly. So it's quite cool research. Actually, I suggest you to Google it or find on YouTube. There are great experiments how scientists understood that they actually use Milky Way. But well, I think they take some specific fraction of light, which is actually sent by Milky Way, so and use that as far as I know. I am not the expert to discuss this. It's not phylogeny or taxonomy. Sorry?
She said the use of the Milky Way could be one of the behavioral characteristics you could look at. So, but I was going to respond to Lou and say that John Weens, who's now at the University of Arizona, has done a lot of behavioral modeling into the phylogeny, looking at these sort of phenotypically varying and also discrete behaviors, and use those in lizard morphological, lizard phylogeny. So I don't know if you know John Weens' work, but that would probably be useful. So my question is, what is your hypothesis about that movable horn? You said you're going to be looking at it. What do you think that movable horn is used for?
Well, one of the most likely and easiest explanation is that it's used for display for females. So just males can move horn and give some signals to females. So probably that's it.
Just another extended sexual selection kind of thing?
Yeah, yeah.
Okay.
So in the beginning, you mentioned that you have classified those beetles using like 200 different features, right?
Mm-hmm.
I'm not an expert on I guess features of beetles, but I wonder how person-specific description of those features might be. That is, if somebody else takes the beetles that you analyzed and doesn't, well, maybe studies beetles, but as well, how unique are these features as you're saying? So are they blindly analyzed? You don't know what beetle is coming from? Is it all quantified in some ways? That the sizes are very consistent, etc. Because I can imagine it's different ages, different, maybe somewhat developmental developmental stage may be the same. So how robust is that data?
Actually, it's a good question. What is the source for subjectivity here, right?
Or
if
objectivity, actually. I think there is a lot of subjectivity. I wonder how much objectivity is there.
Yeah, actually, it's a good question.
Who else looked at that data and recreated that? So can we trust those 200 characteristics?
Yeah, it's actually this procedure of scoring morphological characters. It's totally subjective because you normally work alone with the collaborators, and there are no methods to avoid this subjectivity because actually different science can have different opinions. And the stumbling block here and the associated problems arise because of two factors. First of all, different scientists can have different hypotheses of homology. For example, one say no, this leg is the same with that leg, and the other says no, they are totally different and non-homologous. On the top of homology, there might be various ways how to score a particular character, even when homology is certain, into character matrix. Because basically, you have your data in beetle, and then you need to formalize them into character matrix, right? You need to score somehow. So, and these sort of actually, the methods for scoring are also subjective. One can come up with multiple ways how to score particular characters, and there is like no specific procedure to do that. So yes, it's subjective, but you can go and reinvestigate that, yeah. But I mean, there is no other way. And well, but of course, definitely, if different scientists score the same organ, the same species into metrics, the result will be a bit different. But in general, there are some, patterns which will be common between both of them. So that's why also we use molecules to infer phylogeny. But of course, it's basically new methods have to be introduced into biology to avoid this subjectivity. It's possible, but it's complicated to do that.
So on that topic, I just heard jackknife, right?
I was wondering if you could jackknife out certain labs and then just see how much it changes your model. I mean, there might be a...
Yeah, you can. I mean, you can calculate support, and I just didn't show that here. So, but yeah, it was done in the paper. So you can also, as I said, there are some techniques in parsimony analysis where you can kind of elucidate those characters which introduce phylogenetic noise. So this can be done, but the main source of subjectivity, of course, in morphology is the scoring procedure, because different people see different things in the same organism.
So a question about the dating, right? So I mean, I know there's lots of coprolite fossils and things like that from Cretaceous. I mean, can you use any like trace information from those? Like, can you find evidence of feeding by dung beetles on coprolites, or is it?
Yeah, actually, as I mentioned, there were two ancient fossils from Asia, two oldest fossils which were assigned to dung beetles from Upper Cretaceous, but actually they turned out to be not dung beetles. So. And
There's
coprolites.
body fossils, but there's also like coprolites.
Yeah, coprolites. Yeah, there are
dung,
coprolites. There is one finding. Well, there are many coprolites, but there is actually one specific finding with tunnels. So not with the remains of dung beetles, but with a tunnel, with a trace of tunnel, which is filled with some maybe organic matter. So, and actually it was claimed to be the trace of dung beetle, so that the oldest dung beetle actually arised in that period. So, but you can't judge from this tunnel because there were other scarab beetles, not dung beetles from the subfamily Scarabiini, but from the other subfamilies, which are older. So, and they also dwell in dung. So it well might be that it was, for example, that another beetle, or maybe something else, some other invertebrate, who knows. So it's quite unreliable, I would say.